Hello, I am Professor Somode. I am an assistant professor in the School of Bioscience at IIT Kharagpur. Today I will talk to you about gel electrophoresis of DNA and proteins. This is a two-part lecture, and in this part I will talk about the basics of gel electrophoresis, and in the next part I will talk about some of the applications of this method. So, gel electrophoresis is a very versatile technique that is used in almost all biological labs. So, we will specifically focus on two types of gel electrophoresis. One is the SDS page or the page polyacrylamide gel electrophoresis, and the other one is the agarose gel electrophoresis. We will see how this technique works and under what conditions you will use which method. So, for polyacrylamide gel electrophoresis we will see two applications, one under denaturing condition SDS page and the other one native page, and we will talk about the separation principles, how these macromolecules are separated in this gel electrophoresis, and also I will try to talk about some of the practical aspects of this technique. So, typically, so these are some of the typical features of gel electrophoresis that are listed here. So, there are two phases, one is the gel matrix, which is the stationary phase, and another is the liquid, which is the mobile phase. So, liquid is basically the buffer that we use, which has various ions. The gel matrix is made up of some polymer. In case of polyacrylamide, it is the polyacrylamide polymer, and then the other one is the agarose polymer. So, this gel acts as a three-dimensional molecular network, and this molecular network has various size of pores through which these molecules can pass. So, the gel matrix acts actually as a molecular sieve. It also prevents convection in the liquid phase and also lowers the rate of diffusion of the molecules. So, on the right hand side what you see is a rectangle. So, a typical gel would look something like this. The thickness of these gels are very small. If it is a polyacrylamide gel, this thickness will be something in the order of 0.5 millimeter. If it is an agarose gel, then it can be up to 5 millimeters. So, what we do is we apply an electric field across this gel. So, one side is a positive terminal and the other side is the negative terminal, and these molecules are ions. So, the negatively charged ions will start moving from the negative terminus towards the positive terminus, and what we will also see is that inside the gel we have certain lanes. So, that the samples are applied at the top in these lanes, and when the electric field is applied they run vertically in this direction. The presence of this gel reduces diffusion across the lanes. So, there is no mixing between the samples across these various lanes. Okay. And also the pore size of the gel determines the distribution range of the macromolecule that can be separated through that gel. So, as I mentioned before, gel electrophoresis, there are two types. We are going to talk about, there are many, but in this lecture I am going to focus mostly on two types. One is polyacrylamide gel electrophoresis, the other one is agarose gel electrophoresis. Polyacrylamide gel electrophoresis is used to separate large biomolecules such as proteins and DNA, and agarose gel electrophoresis is used to separate even larger biomolecules, and in this case it is typically DNA and RNA. So, page is used mostly for proteins, and it is used for DNA if the length of the DNA is less than 200 base pair. If the DNA or the RNA is more than 200 base pair, then we have to use agarose gel electrophoresis. Okay. So, what is the basic principle by which these macromolecules are separated? So, typically what is done is that all these macromolecules are given a negative charge. So, in case of DNA and RNA, they naturally have negative charge because of the phosphate backbone. But proteins, depending on the pi of a particular protein, can be negatively charged or positively charged based on the buffer of pH of the buffer that is used for electrophoresis. So, in case of DNA, the DNA or RNA, the molecules are negatively charged, but in case of proteins, the molecule is made negatively charged by adding some chemicals such as SDS, sodium dodecyl sulfate. So, in essence what we have is a charged particle and then an electric field is applied across the gel. So, this charged particle experiences an electric field. So, this electric field will push the molecule towards a positive or a negative terminal. If it is a negative charge, then it will start moving towards the positive terminal. Now, these gels have certain pores. So, when the molecules are passing through these pores, they face a friction. Okay. So, there is a viscous drag which acts in the opposite direction of the electric field of the movement of the proteins, and this viscous drag is proportional to the size of the molecule. So, the velocity of the molecule is given by this equation, where E is the electric field, Q is the charge divided by F, which is the frictional coefficient, and this frictional coefficient depends on the shape and mass of the molecule. So, typically these experiments are done at a constant voltage, which means that the velocity of the molecule depends on the ratio of the charge and the frictional coefficient. Okay. Now, if we think of DNA, it is easier to think DNA molecules in terms of this gel electrophoresis. These are typically long molecules. All DNA molecules have very similar shape, and the charge distribution is also exactly the same for all DNA molecules. So, it turns out that they have similar shape and they have similar charge to mass ratio. So, under these conditions, these molecules move depending on their size. So, only the size is the one that determines whether a DNA molecule will move faster or slower. Smaller molecules will move faster because they can pass through smaller pores as well as the larger pores, and the larger molecules will move slower because they can pass only through the large pores and not the small pores. So, similarly, we will see that we actually use a trick to make uniform charge to mass ratio for proteins by using a molecule like SDS. So, the first gel electrophoresis that I am going to talk about is polyacrylamide gel electrophoresis, or in short it is called PAGE. Okay. So, this gel has smaller pore size compared to the other one, the agarose gel, and hence it has a very high resolving power for biomolecules up to 1000 kilodaltons. If the biomolecule is bigger than this, then we will have to switch over to agarose gel. So, some of the physical qualities of PAGE gel are listed here. One is that it has very good physical stability and thus is easy to handle. It means that after running a gel, you will have to take the gel out from the glass plates, you will have to stain it. So, a lot of handling is required. If the gel matrix breaks easily, then it becomes very inconvenient. PAGE, even though it is very thin, a very thin gel is used, it has a very good physical stability, which means that it can be easily handled without damaging the gel. The sample separation is due to both molecular sieving and electrophoretic mobility. So, molecular sieving is larger molecules will move slower, smaller molecules will move faster, and electrophoretic mobility means that molecules with higher charge will move faster, lower charge will move slower. So, the mobility is because of the charge on the molecule. At this point, it is important to point out an important difference between these gel electrophoresis and the size exclusion chromatography that you have learnt about in previous lecture, is that there are no void volume in the matrix of a gel. So, in this SDS page gel and also in agarose gel, they have only continuous network of pores, which means that in case of size exclusion chromatography, larger molecules can enter the void volume, and so they can quickly reach the end of a ACC column. But in this case, all the molecules will have to pass only through pores, since there are no void volumes. So, small molecules can move through both small and large pores. So, they move faster, and larger molecules move slower. So, this is opposite of what we have seen in case of a size exclusion chromatography. And the pore size of the gel matrix can be controlled by the concentration of acrylamide. Okay. So, we will see that since this is a polymer, we use two types of monomer. One is acrylamide, the other one is N,N-methylene bis-acrylamide. So, by varying the concentration of these two monomers, we can change the size of the pores. So, the chemical structure of these two monomers are listed here, acrylamide and bis-acrylamide. So, what acrylamide does is, if you polymerize this, then polymerization happens across this double bond, and what we will get is a linear chain of a polymer. Okay. But this linear chain of a polymer will not give us a three-dimensional network. To get a three-dimensional network, what we need is this bis-acrylamide. So, bis-acrylamide can connect two such linear chains and provide a three-dimensional network of polymer. So, if a student forgets to add bis-acrylamide and polymerizes only acrylamide, then what you will get is a highly viscous liquid, but not a solid gel. If you see carefully, bis-acrylamide is nothing but this acrylamide moiety. So, there are two acrylamide moieties, this and this, and these two moieties are connected by a methylene group. So, that is why it is N,N prime methylene bis-acrylamide. And as I mentioned before, the pore size of a molecule of a gel can be controlled by varying the concentration of these two components. So, typically acrylamide is used somewhere in between 4 to 20% weight by volume, and bis-acrylamide is used somewhere in between 1 to 5% of the concentration of acrylamide. Okay. So, if we use higher concentration, we will get smaller pore size. If we get lower concentration of these two, we will get larger pore size. And also one caution, these monomers, the monomer acrylamide is neurotoxin, and it is also suspected to be a carcinogen. So, one has to be very careful while handling the monomers. Once it polymerize into a gel like this, then it is not harmful, but the monomers are harmful. So, we have to be careful when we are handling the monomeric acrylamide solution. Okay. So, the advantages of polyacrylamide, so there are certain advantages. One is that it does not interact with proteins or nucleic acid. So, there is no reaction, it is a very inert medium. It is chemically stable, hydrophilic, and also free of ions, which means that all the electrophoretic mobility that we get is because of the ions that are either supplied in the biological sample or in the buffer. This gel itself will not contribute any ion in that electric current that passes through the gel. Also, it does not interfere with the common staining reactions. All these solutions are colorless. So, at the end of a gel electrophoresis, we have to stain the gel in order to see the protein or the DNA. Now, this polyacrylamide gel, it does not interfere with the common staining methods that are used, which means that only the protein or the DNA bands are stained and the rest of the gel remains clean, which makes it easy for us to detect the protein or DNA or RNA bands. So, now we are getting into the details of this gel electrophoresis. What I am going to talk about now is the is something called SDS page denaturing gel. So, in this case, as it says denaturing, it means that, so this is typically used for protein samples, and denaturing means that we are going to denature the proteins and run it onto a gel. This method is very useful if you want to check the purity of your protein sample, and also if you want to estimate the molecular weight of your protein sample. If the molecular weight is not known, you can actually determine it from this method. So, in this method there are certain features that I would like to point out. A typical gel would look something like this. You can see there are two gels. The upper one is called a stacking gel and the lower one is called a resolving gel, and these are called the lanes. So, this is where the protein samples is applied. So, in this setup we can actually apply 10 different samples and run it simultaneously on the gel. A typical experiment will be will have 9 samples in this 9 lanes, and the first lane will have a molecular weight marker. So, it is a mixture of standard proteins with known molecular weights, and when they run here they will show up in different places, and from that we can actually estimate the molecular weight of other unknown proteins in these different lanes. So, we can make a direct comparison between samples in each lane. So, as I mentioned before, there are two gels which are stacked on top of each other. The top one is the stacking gel, the bottom one is the resolving gel, and we use 3 different buffer systems in this discontinuous gel electrophoresis. The stacking gel is prepared using a buffer of pH 6.8, the resolving gel is prepared using a buffer of pH 8.8, and the running buffer, so this is the buffer which actually passes through the gel and is responsible for the electric current that goes through the gel. This buffer is at pH 8.3. So, we have 3 different buffers with 3 different pH values. The stacking gel has a very important function. So, the resolving gel, as the name suggests, is the one which resolves all the protein molecules that are as a mixture in a particular sample. So, suppose in this particular lane we have 5 different proteins, and this resolving gel will resolve them into 5 different bands according to their molecular weight. The stacking gel performs a completely different function. So, if you imagine a race where 10 different athletes are running, so to start that race what you have to do is all the athletes should come to the same starting line, and when the gun is fired all of them should start running simultaneously. Then only we can say who is running the fastest. Similarly, in gel electrophoresis what we need to do is we should have all the protein molecules at the same starting line. So, if this is the resolving gel, then all the protein molecules should be at the same point, but when a sample is applied, for example this one, these samples occupy a certain volume. So, we typically add something in the range of 10 to 15, maybe up to 20 microliters of sample in each of these lanes. So, if this sample volume is 15 microliters, it means that all the proteins are distributed in this volume. So, the protein that is down here, the molecule that is down here will enter the resolving gel first, and the molecule that is up here will enter the resolving gel later. So, there will be a diffused band, and it will be very difficult to tell which protein is running faster and which protein is running slower. So, it is important to get all these molecules in a very small tight region before they enter the resolving gel, and that is the function of a stacking gel. That is why it is called a stacking gel. So, once we run the gel, this is exactly what we would see. If this particular sample has a mixture of these 5 proteins, then these 5 proteins will separate out in that lane according to their molecular weight. The smaller one will be at the bottom and the largest one will be at the top. So, in case of SDS page denaturing gel, there are two more chemicals that are added. One is sodium dodecyl sulfate or SDS, which is a detergent, and the other one is beta mercaptoethanol or BME, which is a reducing agent. So, BME breaks all the disulfide bonds that are there in a protein. Once a protein is dissolved in the loading solution which has SDS and BME, it is actually boiled at 100 degrees centigrade. This boiling unfolds the protein, and once the protein is unfolded, SDS will bind the hydrophobic regions in this unfolded or denatured protein. Once it binds this denatured protein, what happens is that the complete protein is now coated with a negatively charged molecule, because SDS is a detergent which has a long hydrophobic chain and a negative ion. So, it does not matter whether a protein has a low pI or a high pI. At these conditions, all these proteins are denatured and they are uniformly coated with a negatively charged SDS, which means that all proteins have now been converted to negative charge, and it also ensures that all proteins have a constant charge to mass ratio, which means that these proteins will now be separated depending only on their size, because they have a uniform shape, they have a uniform charge to mass ratio. So, some of the chemicals that are used in SDS page gel electrophoresis are listed here. So, this is sodium dodecyl sulfate. You can see that it has this long hydrocarbon chain and this negative charge. So, this hydrocarbon chain binds to the hydrophobic regions of the protein, and this negative charge provides the negative charge, overall negative charge of the molecule. Two more chemicals are used. One is ammonium persulfate and the other one is timid. If you remember the structure of acrylamide, it has this double bond and the polymer is formed across this double bond. So, this reaction is mediated by the formation of free radical, and this free radical is supplied by ammonium persulfate, which acts as a catalyst. So, when ammonium persulfate is dissolved in water, this peroxide bond is broken spontaneously and it forms free radical. But it turns out that the free radical formed by ammonium persulfate is not very efficient in producing free radicals on the acrylamide double bond. So, that another compound is added, which is N-N N-prime N-prime tetramethyl ethylene diamine, or in short TEMED, TEMED. Ammonium persulfate free radicals can easily generate free radicals on the TEMED, and these are very efficient in starting the polymerization of the acrylamide. So, this is the catalyst, this TEMED is the initiator, and together they start polymerization of the acrylamide bisacrylamide mixture. One very common mistake that students make is they forget to add one of these two components. They will either forget ammonium persulfate or they will forget TEMED. In either case, what you will get is that even after waiting for hours, there will be no polymerization of your gel. So, in that case, the only option will be to pour out the solution mixture from between the glass plates, but remember that unpolymerized acrylamide is harmful. So, you should not dump that into the lab sink. What you should do is take it out in a beaker, add some ammonium persulfate and TEMED, let it polymerize, and then you can scrape it out and dispose it. So, coming back to the SDS page denaturing gel, again this is our setup. So, what we have at this point is a stacking gel, a resolving gel. We have added our sample to these lanes, and we have applied an electric field across it. Now, all our protein molecules are negatively charged, which means that they will all migrate towards the positive terminal, and the running buffer is also there, which has a pH of 8.3. So, the majority of the current that passes through a gel is carried by the buffer ions, for example, tris, glycine and chloride. These are the two negative ions which will migrate with the proteins towards the positive terminus. Protein ions, even though they are negatively charged, they have negligible contribution to the current. So, it turns out that the concentration of the buffer is very important. Again, a typical practice in any lab is that we make 10x buffer solutions, and before using or before running a gel, that 10x solution is diluted to 1x and the gel is run. If someone forgets to dilute it to by 10x and makes it, let us say, 20x or even more, then the buffer concentration will be too less. Now, if the buffer concentration is too less, then the current that passes through the gel also results in faster migration of the protein, because if there are not enough ions, then the protein ions also start contributing to the total current of the current passing through the gel, which means that these proteins will migrate fast and it will result in smeared bands. So, we will not get nice crisp bands, and it will be difficult to determine where our protein is. If the buffer concentration is high, so if I just use 10x buffer and forget to dilute it, then the proteins will move very slowly. So, it is important to have the right buffer concentration. So, that brings us to the function of the stacking gel. How does this stacking gel stacks all the protein and brings them at all the protein molecules and brings them to the same point? That is the most important thing. So, this part is the stacking gel pH 6.8 and this part is the resolving gel pH 8.8. The trick is that what we use is glycine. This is the structure of this amino acid glycine. It has a pI of 6.5. So, at pH 6.8, which is very close to its pI, glycine stays in these two different forms. This one is the zwitterionic form, where the carboxylic group is negatively charged and the amino group is positively charged. So, it is neutral, and a small portion of the glycine will be negatively charged. But in the resolving gel, where the pH is high 8.8, almost all glycine will be negatively charged. Now, this is our stacking gel. The chloride ions have very high electrophoretic mobility. So, in this stacking gel, when the voltage is applied, the chloride ions move very fast, and what they do is they actually form the front. So, there is a very tight chloride ion front that is formed in the front of the gel, because they are moving the fastest. Glycine at this pH 6.8, most of it is neutral and very small amount of glycine is negatively charged. So, its electrophoretic mobility is actually quite less, and also the dearth of ions in this region results in an effective increase in the resistance, because we have less ions which can carry the current. Now, remember that the current that flows through the gel is constant, because there is no accumulation of charge, right. So, if the current remains constant, if the resistance increases, then from Ohm's law we can tell that the voltage will also increase locally in this region. So, when the voltage increases in this region, the protein ions start moving faster. And one trick that we use for this stacking gel is that a very dilute acrylamide solution is used, which means that the pore sizes are very large. So, there is no sieving effect. Small molecules, small proteins, large proteins all can easily pass through this gel. So, all of them pass through the gel, through the pores quickly, and since the local voltage is high in this region, they will quickly reach the chloride ions. The moment all these proteins reach the chloride ion, the concentration of chloride ion is very high here, which means that the current is high, right. So, the effective resistance goes down. So, the voltage also goes down. So, these protein ions will stay right behind the chloride front. So, the effect is that all the protein ions are stacked into a very narrow band right behind the chloride ions, and together they reach this intersection and they enter the resolving gel as a nice tight narrow band. The moment this glycine, chloride ions and proteins they enter the resolving gel, the pH of the resolving gel is 8.8. So, now all of the glycine molecules are quickly converted to this negatively charged ion, and they start moving fast. So, this concentration effect that we saw in the stacking gel is now lost in the resolving gel. Also, in the resolving gel, a higher concentration of acrylamide is used, which means that now the sieving effect comes into place, and it will start resolving the protein according to their size. So, once the gel is run, we need to visualize them. So, while the gel is running, all of these are colorless. So, we do not see anything. So, in order to ensure that we know when running the gel is complete, a tracking dye is added, typically bromophenol blue. So, it has negative charge and it has very high electrophoretic mobility. So, it runs the fastest, and once this tracking dye reaches the bottom of the gel, we know our gel electrophoresis is done. And later on, to stain the proteins, typically one of these two or a mixture of these two molecules, Comasi brilliant blue, is used. So, here what we see is a setup of a SDS page gel electrophoresis. This gel is run vertically. So, this is where the gel is run and electric current is provided by this power source. These are the different components. So, this is the tank where the protein, so this is the glass plate, the gel is formed inside this. This glass plate is packed inside this, and this whole thing goes inside the tank, and the buffer is filled into this tank, and then you set it up and pass electric current through it. So, once the gel is run and once we stain it, it would look something like this. So, this is a typical molecular weight marker that is used, where these different bands correspond to different size proteins, and from that we can tell that what is the molecular weight of our protein. So, this protein will be somewhere in between 10 kilodaltons to 15 kilodaltons, and sometimes when we purify protein, we can tell that our protein is highly pure and there are no other bands in this gel. So, we can determine the purity of a sample, and we can also roughly estimate the molecular weight of a sample. So, that is all for this lecture. In the next lecture, I will talk about some more applications of gel electrophoresis and some other types of gel electrophoresis. Thank you.